Okay, so today we will discuss we are starting a new chapter, that is enzyme. It's the chapter number 3 of our zoology portion. Okay, so first of all, let's decode this one term. Enzyme is a Greek word. It is a Greek word which means inside of the yeast. In means inside, and zyme means yeast. Okay, so basically, what is enzyme? Enzyme is a biocatalyst. First of all, let me tell you that what is catalyst. Catalyst is someone that accelerate the metabolic reactions. It is a type of chemical that speed up the reaction. It is a type of chemical that speed up the chemical or metabolic reaction. Okay, this is the definition of catalyst. But what is biocatalyst? As we know that catalyst is something that is speed up the chemical reaction. So biocatalyst is something that is speed up the metabolic reactions that take place in a living organism. For example, we are having let's take the example of Risic. Okay, what is it basically? It is something that it is a type of medicine that speed up the reaction that helps to digest a food very clearly, very in rapid formation. So enzyme is a biocatalyst. Basically, enzyme speed up the metabolic reaction that takes place inside of the living organism. It speed up the metabolic reactions that takes place inside of a body. Okay, so there are two type of energy of activation and activation and energy. So okay, so we will discuss this in the next one video. So uh, during the reaction, okay, during the reaction, the enzyme that remain unchanged. Okay, during the reaction, some of the enzymes remain unchanged, are of proteinaceous nature, but those enzymes that changes itself during the reaction are known as activator. Though that changes itself is known as activator. For example, salt or magnesium. It means that for example, uh, we are cooking something. Okay, we are cooking something and we put salt inside of that dish, and the formation of the salt or the structure of the salt just get dissolved in that one water or in that one liquid. So it means that it changes itself, it changes its structure. So the salt is the activator. It is an enzyme, but it is the activator. Okay, if we talk about the history, so before 19th century, before 19, it was believed that enzymes were only of proteinaceous nature, and they they believe that. And there's a scientist who is named as Thomas Schech and Altman. These two scientists said that RNA also work as enzyme in the form of ribosome, and ribosome is present in ribosome. Okay, so before, but after the 19th century, it is proved that there are two types of enzyme. The, both have uh, uh, enzymes have maybe have proteinaceous nature and non-protein nature. There are two types of nature in enzyme: the proteinaceous and protein nature. Okay, so basically, enzyme is something that catalyzes a reaction or speed up the metabolic activities that takes place inside the body. And the Thomas Sachs and Altman say they believe that before the 19th century, it is a belief that RNA also work as an enzyme in the form of ribozyme and ribosome. But after the 19th century, clarified by it is clarified by some scientists that enzymes have two types of nature: protein and non-protein nature. Then, during the reaction, the enzyme does not change itself, but those enzymes who changes itself are known as activator. Okay, so this is all for today.